So I have the great honor and massive pleasure to welcome all of you here to the launch meeting of ICDA today. We are a group that have come together in the notion that the time is right to accelerate genetic findings into biological discovery and to medical action. Some of you have asked about the genesis of ICDA, and about 2015, a bunch of us realized that we on our own had been sitting in our chambers and thinking about what the next real step is. We have done really well in identifying genetic associations, but the therapeutic opportunity that has sprung out of that has not been as fast. So what could we do about that? And I think there was a realization that there were enough of us that we actually could aggregate and start to talk about this as a group. So in 2018, a handful of us met in New York to discuss whether this was a good idea at all, and whether we should start some sort of working group to think about the best way of sort of really propelling this forward and do a serious effort to address the question. The meeting was successful, we felt, and we decided to invite further stakeholders. So about 90 people met in Oxford in the autumn to follow up, and all these lovely faces were there. And they represented a range of stakeholders from biomedical scientists, from pharmaceutical companies, ethicists, epidemiologists, geneticists, and so forth, all thinking about common human disease and what we could really do to accelerate progress together. So we decided it was a good idea to come together and form an alliance, an international alliance, to sort of address the challenge. We decided roughly how we wanted to work together, so we formed an organizing committee, and the organizing committee met in Helsinki to start to really think about the most important key scientific questions. And all of you have received the white paper draft that we have started to put together, so I'm hoping everybody has at least glanced through that. And really sort of starting to think about different working groups and different questions that we wanted to address. So what you see here is the organizing committee, which consists of 33 members from 5 different continents. And here today, I think we're close to 170 individuals from 19 countries spanning 6 different continents. And that reflects the ethos that we really, really want to capture. We want it to be an international effort. We want it to be inclusive. We're thinking about, diversity of all different axes in the working group, with the notion that when we come together and when we amalgamate our thoughts coming from different working groups, coming from different stakeholders, we're going to come up with the best solutions together. So I'm really excited to welcome you today. I'm encouraging everybody to just feel like it's relatively informal, so you can speak up and share ideas and raise questions during the days that we're going to spend here together. And I'm going to talk together with Eric and Matt here today, and we are going to talk about the past, the present, and the future, setting the stage, if you want, for the discussion we're going to have today. So I have the pleasure of talking on the past, way before my own career in genetics started. And starting out, we were all really excited about finding the gene for common diseases. We had looked at monogenic disorders where Dr. Collins and other people in the audience had worked really hard to find markers that co-segregated with disease in families, doing so-called linkage analyses, and identifying regions that likely contain a disease-causing gene or a mutation in a gene that contributed to disease. That has been incredibly successful for monogenic disease, but less so for common complex disease, where there is only really a handful of genes that have been identified that way. So we were partly hampered by the very early genetic maps. So I still remember doing microsatellite genotyping with fluorescence, but I know that there is some in the audience who've done that radioactively, not looking at you, Mark McCarthy. But that is time-consuming with low throughput, expensive, and you can't really do that in high throughput. So there was this really seminal paper that I read, and many others did, from Neil Risch and Kathleen Mary Kangas that came out in Science in 1996, discussing how for common disease, where you would expect a relatively small effect sizes, and I would say that the ones that they put forward in their simulations are actually quite large, but linkage analyses wouldn't do so well. So we should do association analyses, and that would be much more efficient. For those of us who worked in the field of association knows that that was cluttered by inconsistent results with heterogeneous interpretation of results, and it was a little bit all over the place, though we all did our very best to do good science. I think we learned from the association studies when we started to look at it in aggregate and perform meta-analyses that most candidate genes were not reproducible due to power. There were genes that were selected based on candidacy, ascribing too far importance on what we felt was the best candidate gene rather than searching through the genome. And also thinking about heterogeneity in terms of population stratification, genetic effects that differ in different sexes and so forth. And that we should expect low effect sizes, heterogeneity, and that we need to aggregate into groups if we're going to get robust results. So when the Human Genome Project sprung out and we got the blueprint for what is literally all of the human biology, we started to glean into the massive amount of human variation that we actually have at our fingertips that could be useful for this. People started to talk about common diseases might actually be caused by common variants, and it sounds really simple today, but when it was put forward, it was really sort of a groundbreaking, seminal sort of thought. The SNP consortium very quickly sprung out of the Human Genome Sequence Project and also provided the first map of human variation and showed us that about every 300 base pair we have a common variant, which is much more than many of us had anticipated. Another observation that was really seminal here was that we started to see that variation was co-inherited in blocks, so-called haplotypes, and that the haplotypes are relatively simple and relatively common, and that's actually something we could utilize when we're designing our studies. So out of the SNP consortium and the Human Genome Project sprung the HapMap consortium, the International HapMap Consortia. And here the realization was that if you have a relatively small number of markers that is informative for haplotype, you can drastically lower the dimension of genotypes required to really get a good coverage over the genome with something called imputation. In parallel, the 1,000 Genomes Project had set out to characterize all the common variation down to about 1% across population, and people started to really come up with tools for analyzing population structure. The third sort of breakthrough technology here was that because we could reduce the space for genotyping, suddenly we could genotype the entire genome in an array, something that wasn't possible before. And that really set the stage and ramped up for the genome-wide association study. So the advent of the genome-wide association study came in the first paper that I could find. I'd done a Twitter survey, and Twitter agrees that Osaki 2002 from Riken is the first GWAS that was published. It's a myocardial infarction case control studies in 1,000 cases and 1,000 controls that report 2 loci. And that was followed by a paper in Science in 2005, which was the first one I read, but where they only used like 96 cases and about 100 controls or so. So remarkable that they found an association, but given the odds ratio of the SNP and the association to it, they had this remarkable finding for age-related macular degeneration. So that sort of gave us a glean of the sample sizes that were needed. The Case Control Consortium from the Wellcome Trust came out in 2007, looking at 7 common complex disorders and 3,000 shared controls. And many people saw that as a medium success, because there weren't as many associations as we had expected initially. And I remember especially the hypertension group, which you will see here in the middle, were completely shocked when they found no associations. And that again led us back to the linkage days where people had started to aggregate. Nancy will remember the first linkage consortium that we had in type 2 diabetes, and the IBD world will know that they also had a linkage consortium. That we need to aggregate together. We need to come together. We need to pool data together in order to increase power. And when we started to do that, we became really, really successful. So today, looking at the genome-wide association catalog, there are over 60,000 genome-wide associations reported with over hundreds of various traits. So I think we can declare some degree of success for genetic mapping. But we shouldn't tap ourselves on the shoulders too much. Over 80% of the GWASs that have been put forward are from Northern European descent samples. So we need to do better to capture global diversity. On top of that, there is a large unexplored area in sexual dimorphism. There are tons of diseases and disorders that don't have a GWAS for a range of reasons, though that's coming in the biobanks very quickly now, so that's a wonderful resource. We have only started to scratch on the surface of really rare variant association, which is something that is really exciting. And I think that's just setting the stage for the challenge. Like, how do we best propel these associations into function, into mechanisms, and how do we take it forward for medicine? So I think we build on a treasure trove of genetic association here to really propel ourselves forward, but there is a lot of work to be done. So I'm going to hand over to Matt here, who's going to talk about the present. So we're going to do a seamless step over.
Thank you very much, Cecilia, and welcome to everyone. It's an incredible opportunity to bring all of us together to really think about the future, and we've got 2 days to really make the most of it. So I get the joy of telling lots of experts in the room what they already know and what they've already done. But what we know is that we have collectively made over 60,000, as Cecilia said, associations, and that's grown very rapidly in the last few years, although that axis has gone rather strange very quickly. But also, we're not just making new associations and many more associations in the same traits. The number of traits for which we have associations is also growing somewhat more organically, as shown on the right here. And what I'm going to try and do over the next quarter of an hour or so is just distill out some of the key lessons that we've learned out of that, especially lessons which we would anticipate will translate into how we take things forward in the future. So the first thing that we know is that of those many tens of thousands of associations, well over 99% of them are for common variants. And what I'm just showing you here is a couple of examples of those rare variant associations, which we know exist in many, many large numbers, but we haven't really had the power to interrogate. And the first one is from exome sequencing in Latino populations, identifying one particular variant within HNF1 alpha that has pretty impressive odds ratios for type 2 diabetes. And the second one is a different type of rare variant association, pooling variants with a similar functional effect, in this case truncating variants, across the SETD1A gene and increasing risk of schizophrenia very substantially. So there's two different flavors of rare variant associations that we have the opportunity to see much more of in the future, because they really are way less than 1% of the associations we found thus far. Another lesson that we have learned and that Cecilia alluded to is the importance of studying different populations, and especially special populations that by dint of their population history are particularly informative. And I give one example here of beautiful work in Greenland identifying a very high effect size variant at actually very appreciable frequency, 17% allele frequency in Greenland, again conferring risk to type 2 diabetes. We have a lot to learn from these variants, and we need to characterize these populations to learn those lessons. We also know that the vast majority, unlike the 3 examples I've given you before, which are all in the coding sequence, those are the exceptions to the rule. The vast majority of these associations are unambiguously affecting gene regulation. And here's one nice study on blood cell traits where, with over 1,000 GWAS on a set of traits characterized in a consistent manner, one can see that in orange we have the annotations of protein coding genes that contribute increasing amounts as variant frequencies go down, the associated variant frequency goes down. But the vast majority of our associations we have in those 60,000 are in that right-hand bar there, where only a small minority are protein coding. So we have a huge challenge to understand gene regulation. We know that the traits that we're studying are incredibly polygenic. The study on the left is one example showing if one identifies the top 1% of the genome that's associated with a given common disease, that accounts for actually, for most common diseases, a tiny fraction of the overall heritability. One has to reach out into 10%, 20% of the genome before one can start to account for the majority of the heritability. And that's led to new conceptual models as to why the genetic risk is so dispersed amongst the genome for most common diseases. Those conceptual models haven't reached any kind of consensus yet, but there's clearly a fertile debate about which will continue. That polygenicity leads us on to different ways that we can use these association signals above and beyond identifying individual associated loci. And polygenic risk scores, which to my mind came to my notice with this beautiful work in schizophrenia, showing that aggregating the association signals across a very large fraction of the genotyped variants, in his case for schizophrenia, identified a signal that could replicate across different schizophrenic cohorts, other psychiatric diseases, but not at all across non-psychiatric diseases. And then fast forward less than a decade, and now we're talking about the potential clinical utility of these, of identifying individuals who have absolute risks that are equivalent potentially to monogenic risks, which are already in clinical practice. So one of the opportunities we have is to think about the biology that's being revealed by these different associations that we find. And one of the key observations is that the biology that's being identified is often very consistent between those monogenic, very high effect alleles that are identified in related traits and the polygenic, weak effect alleles. And this is one nice analysis showing that where polygenic signals and polygenic signals overlap, the squares on this diagram, the vast majority of them are in traits that are clearly related to one another. So this kind of comparative analysis across traits becomes incredibly informative in identifying a general rule that we're seeing the same underlying biology being identified by completely different study designs in different sets of people, but with a common core biology at its heart. One nice example of how one can see this in the context of GWAS studies and related traits is this association study showing multiple associations for stroke and different subtypes of stroke that also affect various other vascular traits throughout the body. And what we're continually identifying over and over again is associated variants aren't just associated for one trait or disease, but actually are quite frequently associated with many traits or disease. In this situation, they're a trait associated with a set of related traits, but the opportunity with having biobanks, large biobanks with genotyped individuals with very broad phenotypes is to ask those same kinds of questions in a much more unbiased manner and identify unexpected, unanticipated connections between different traits and diseases. The association signals that we generate are not just of interest to geneticists, but also to epidemiologists and the ability to make causal inference. And this is just one example of showing how genetic instruments for high BMI, when screened across all the phenotypes in UK Biobank, seem to suggest that high BMI has a protective effect on various psychosocial traits, such as nervousness. And so the epidemiological community is using these as well as much as the geneticists and the functional genomicists. So there is a broad constituency of stakeholders we need to consider when thinking about moving forward with this next phase of common disease genetics. Now, one of the reasons that some of the people in the room come from the pharmaceutical community is kind of captured in the next couple of slides. The first is that we have considerable numbers of targets for many of these common diseases that are being prosecuted by different drugs, and many of those can be recapitulated by the association signals that we identify. And here, one nice example in rheumatoid arthritis. But there are many, many more association signals than there are drugs for many of these common diseases, so the opportunity space is very large. And the value for actually taking up that opportunity comes from analyses done by various different groups that have shown that genetic evidence for association can be powerful in terms of determining the likelihood of success in clinical trials of particular drugs to targets that have that kind of genetic support. Now, to try and make the most of these association signals, we have to understand the biology. And the biology starts with moving from an associated haplotype to try and identify the causal variant. And considerable progress has been made in most of the common diseases where there are hundreds of association signals, with substantial increasing fractions being identified down to single individual variants. And here, one example in IBD with over 40 variants that have been fine mapped down to a single causal variant, 40 different mechanistic hypotheses to be interrogated. Now, much of that mechanistic insight has come from foundational resources. Foundational resources such as ENCODE, the Roadmap Epigenome Project, and GTEx. Those have been critical in sponsoring a kind of explosion of different tools, different innovation, competition around different tools that sit upon a common framework of powerful resources. And that kind of ethos is something that we need to think about carrying forward into the next phase as well. One of the opportunities that we have with the fact that most of these associations fall in the non-coding space is that non-coding functional elements are typically less active in fewer tissues than genes. So that gives us an opportunity to dive into interrogating what are the relevant cell types for a different disorder, and using the association signals again in ways that we hadn't necessarily anticipated at the start of this journey that Cecilia articulated. But here showing that the schizophrenia identifies CNS cell types, rheumatoid arthritis, blood and immune cell types, and highlights a variety of cell types, including musculoskeletal cell types. So these are leveraging those functional resources that we have at our fingertips, but typically at the level of tissues rather than individual cell types, and there's a huge opportunity there in the future. So some of the kinds of biological insights that have been gleaned have been about identifying particular tissues or cell types that are particularly relevant for disorders, changing the focus of some drug development programs towards looking at cell types and tissues that maybe had been underexploited. But also in individual biological processes, and some examples here about autophagy or synaptic pruning, or indeed the complement system in that first association that Cecilia described for age-related macular degeneration. So there are biological insights that can be gleaned, either by virtue of these kind of large-scale analyses of these fundamental resources, or in many cases by researchers going hard yards and doing the functional follow-up of fine-mapped variants. So a combination of the two has kind of taken us to where we are now, but clearly we need to go at much greater pace to get functional insights for all of those tens of thousands of associations. So in addition to those kind of scientific insights that we've gleaned over the past 15 or so years, there's a set of kind of operational insights about how have we got here that it's worth reflecting on. The first is that analytical innovation matters. Many of the kind of insights that we've gained have been using association signals in ways that we haven't previously thought of, or generating computational resources like imputation servers that enable and democratize the ability to make the most of data. Somewhat ironically, one of the areas that's where it's been peculiarly resistant to analytical innovation is actually detecting SNP associations themselves, where the fundamental methodology we use now differs very little from what we used 15 years ago, despite the fact of many bright people having many good ideas. The key insight from this is it's the power of the data that's led to those insights rather than the analytical method. It's been the scale and the collaboration that's brought those data together that has been critical to making robust associations. The second kind of operational insight is that a lot of insights have been gained from comparative analyses across different diseases, one such example here, and data sharing has been at the heart of this. The data sharing ethos that came from the Human Genome Project has kind of transmitted through this large-scale genetics endeavor has been very positive, but it's far from ubiquitous. It's nowhere near where we need it to be. Summary statistics are not shared on all GWAS. They're not collated in ways that enable them to be used. And we as a community have an opportunity to tackle this. There's been a lot of analytical innovation around in making insights possible without necessarily sharing individual genotypic data. But what we need is comparative work on making sure that those data are actually shared, those summary statistics are shared. And the third is that we've come together in interesting and different ways to try and do this, and that's often involved the public-private partnerships, even starting with the SNP consortium that Cecilia mentioned. And some of those public-private partnerships are around generating new data sets, most recently the announcement of the whole genome sequencing in UK Biobank, for example. And sometimes they're about actually aggregating the data that we already have in ways that make them more easily to port into pharmaceutical companies for it to become an integral part of what they do. And this is just one such example with the Open Targets initiative, bringing together the EBI, Sanger, and a number of different pharmaceutical partners to try and collate together all the data that we've generated to make it more useful. So we have at our fingertips some really important, really exciting opportunities for genetics to influence healthcare. And I tend to break them down into three kind of areas. The first is to transform how drugs are developed. The second is to make sure we're giving the right drugs or the right interventions to the right patients. And the third is to actually give people an opportunity to prevent their disease. And each one of these has challenges associated, challenges that many of us in this room have a responsibility to address. And I've enumerated some of them here, and Eric will go on to discuss some more of these, but we really need to, for developing new drugs, we need to be able to get mechanistic insights for every single one of those common disease associations. We need to know the target gene, the direction of effect, the cell type, the cellular program in which it's operating. For the personalized medicine, the stratified medicine, one of our major challenges here is that almost all of the associations we've done are on susceptibility of disease. They're not about progression of disease. They're not about clinical outcomes. We really need to work hard to get that richer clinical data to enable us to transform our understanding of disease progression in the same way that we have disease susceptibility. That will require patients sharing more of their information, and it will require us to earn their trust to do so. And data security and patient privacy are going to lie at the heart of that. And then when we come to preventative medicine and the opportunities around polygenic risk scores, we have one major challenge, which is that, as Cecilia pointed out, the vast majority of these association studies are done in populations of European ancestries. And those polygenic risk scores work much better in those populations. And we risk a very serious risk of enshrining some major population inequalities in preventative medicine if we don't address this imbalance right now. The second is, if I think to how genetics already operates in healthcare, for example, in diagnostics, there are sets of standards and guidelines and infrastructure that we simply don't have for example, the clinical implementation of polygenic risk scores, that if we're envisaging them becoming of clinical utility in the near future, we need to work collectively together to develop. So there's a set of challenges as I see them, but the opportunities are great. And now I'm going to hand over to Eric to give us a sense of how we can start to address and tackle these challenges.
HDMI?
I think you can. You have HDMI.
Great. Okay. This one is where? Okay. Great. Okay. Let's try this one here. Okay, fine. Two. Sorry, I can do that. That's fine. All right, he's going to show the slides. Excellent. Well, first, thank you to Cecilia and to Matt and to everybody who has come here. This is a working meeting. It's the first meeting, the first public plenary meeting of this nascent ICDA. And while we're going to have a spectacular set of talks, and I'm amazed at the program that we have assembled for this ICDA, which didn't even exist officially until this morning, this is a meeting where our real goal is to pick up on the themes that Cecilia and Matt have talked about, to ask, where are we going and what do we have to do to get there? So in addition to scientific discussions about particular talks, we want to start the process of figuring out what are the plans that are needed going forward to make sure that the next phase of human genetics delivers as well as the previous phases of human genetics. For that purpose, I am delighted that we are here near the NIH and that the NIH is a co-sponsor of this meeting, because I know the NIH is thinking a lot about those questions. There was a time that one could say, well, this was just an issue for the Genome Center and then the Genome Institute, or that it's just an all of us kind of question. But in fact, it's a question that cuts across anybody who's interested in studying disease today. So we're thrilled to have the NIH as partners, and we're going to have a panel of representatives from the NIH talking about their views of it. We're going to have many other discussions that are built into this meeting to talk about where we might be going. And that's all in the context of this initial draft white paper that about 40 people have been working on, but still has a big gaping hole at the end, namely Chapter 3. What are the most important things to do, and how do we go from broad directional statements like, for example, we desperately need non-European populations engaged in all of this, to how are we actually going to do it? It's not enough to say it needs to be done. It actually needs to be a sentence not in the passive voice, but in the active voice. Who will do what about it? By when? Those are the kinds of questions that have been part of human genetics since the 1980s and will carry us forward. I want to make a thank you to Rachel Liao, who is the executive director of ICDA, without whom this meeting would not exist, and without whom an awful lot of the work of the organizing committee would not exist, and her scientific contributions and her executive contributions are much appreciated, and I want to thank her and her staff. So with that, my job in the past, present, future was to do more looking ahead. We're obviously overlapping in these handoffs, but my primary focus will be on what has to be done going forward. But I will do 3 or 4 slides simply to again remind us about phase 1 for the purpose of really asking what worked in phase 1 that we have to do in phase 2. So for Matt and Cecilia, you've heard that one key aspect was to have clear concepts, very clear concepts. The idea that for Mendelian diseases, we could do it by linkage mapping. And if we had a dense enough linkage map, we could pin a single gene disorder down to the chromosome. Whereas for common diseases, the idea was developed that you could do it essentially by tracing not linkage in a family, but chromosomal segments, ancestral haplotypes in a population. That is association analysis. So these were intellectual ideas, but they weren't operational until an awful lot of infrastructure was built. So the Human Genome Project had to be done before one could seriously apply the Mendelian mapping paradigm. We actually had to have genetic maps across the genomes, physical maps across the genomes, so that one could get to the region in which one that contained a human disease gene for a rare Mendelian trait, and then one just had to have the whole sequence so that you could double click on it and pull it up, as opposed to spending years trying to find the gene. Anyway, it got done. It took quite a lot of work by groups all around the world, and it got done. In effect, that gave us what we needed for Mendelian disorders, but not what we needed for common disorders. So we needed, never got this name, but I'm just going to call it the Human Variant Project. The analogous thing that said it wasn't enough to have a few thousand genetic markers up and down the genome. We actually needed millions of genetic markers. We actually needed all of the common variation of the whole human genome if we were going to detect these ancestral segments and do association studies. So we needed massive catalogs of all common variants, everything above frequency of 1%. Now we're actually down to 0.1%, where we have pretty good coverage in European populations. Footnote again, not in non-European populations. We needed to know the haplotype structure of the genome, because we weren't going to genotype 10, 20 million markers. We needed to genotype a subset of markers and infer the rest using imputation methods. But that required understanding the haplotype structure of the genome, the International HapMap project. And then genotyping. Some people in the room remember radioactive genotyping, fluorescent genotyping of single markers, and it was an amazing innovation to get to tens of markers, and then hundreds of markers, and then we all knew that we had to get to millions or hundreds of thousands of markers at a time, and it got done, and we could do that, and then it can now be done extremely cheaply. Those initial studies cost thousands of dollars per sample, and now they're in the tens of dollars per sample. And then, of course, we needed rigorous analytical methods because it was very clear that most association studies resulted in artifacts for lack of rigorous methods, understanding what constituted a significant result, for example, what revealed population substructure, creating artifacts. And the field came together to do all of those things. It all got done. Again, I won't dwell on how it happened, but we went in a short period of time from having 4,000 human genetic markers to having 10 million and now 30 million such things, from having a few examples of haplotype structure to having haplotype structure across the whole genome, and from having Eppendorf tubes to genotyping arrays. And it has given rise to this explosion that my colleagues have already talked about of more than 70,000 such associations across the genome. And it has given rise to lots of amazing biology. All these things were not understood before the era of common disease genetics. Autophagy and inflammatory bowel disease, those words had never appeared in the text of a paper together until after association studies. The fact that HDL was epidemiologically correlated but not actually causally protective was something that was not known and that in fact had consumed billions of dollars of pharmaceutical investment that I bet they wish they had known in advance. Or that non-lipid pathways constitute something like 80% of the loci in the genome that are associated with heart disease. That synaptic pruning plays a role in schizophrenia, that Alzheimer's is probably not primarily a disease of neurons, but something that's acting in microglia. These are just amazingly important physiological observations. The point of age-related macular degeneration and its relation to the complement system, no amount of candidate guessing had thought of that, but the genetics was happy to tell us that. That atrial fibrillation also involves sarcomere proteins in the contractile process and might be, at least in some of its aspects, related to a kind of mild cardiomyopathy, not really thought about. Sickle cell complications, that one can find genes like BCL11A that control fetal hemoglobin, that act as partial modifiers of sickle cell disease, or sets of regulatory factors involved in thermogenesis genetically linked to body mass index. Why did it work? It worked. Well, there were a lot of secrets of success. There was clear scientific direction. We invested in foundational resources. It was not enough to say, everybody do their project. We had to come together and say, what do we have to do that all of us need, but is too big for any one of us to create? We built comprehensive data sets, and comprehensive meant comprehensive. It truly meant we were going to know all the genetic variants, for example, and all of those linkage disequilibrium relationships. We built powerful experimental tools to be able to genotype inexpensively, for example, or now sequence very cheaply. We built data platforms, even in those primitive days of the 1990s and early 2000s. In the pre-internet days, we built the best tools we could so that people could be working on the same data platforms. And critically important to that, we worked out how to do data sharing. I think this field set the bar for what can be done. And it's really clear that had data sharing not gone on, we would have 1,000 separate data sets that couldn't be integrated, and we would be so much poorer. And instead, we built a coordinate reference frame on which everything could be combined. We had flagship projects and many individual projects as well. The flagship projects helped us understand together what were these solutions that really worked and exposed possible ideas to the scrutiny of sunlight to say, does it really work? Everybody picking at it and asking where were the problems. And most importantly, it was an international collaboration. All of this work from the Human Genome Project onward could not have gotten done but for international collaboration. The ideas that came from different perspectives were so important. And when you're talking about the human population, you simply can't study the human population without involving the human population, which means inherently it must be international. So these were the secrets of success. I put them up because we have to keep them in mind as we go forward to a phase 2. There's lots to celebrate. We understand lots of things, and Matt has done a wonderful job of describing the view that we have of the genetics of common disease that even a decade ago we didn't know, and 15 years ago would have been largely rejected, but has clearly turned out to be correct. But the challenge is, despite heroic examples of taking an association and converting it to a gene and studying it and its physiology, it's still more often than it should be heroic examples. It's not like turning the crank. We have 70,000 associations. We'll have hundreds of thousands of associations. It's time, like in every other aspect of this field, to ask, how do we do this in a more systematic way, in a rapid way? How do we build those tools so, well, it used to be one laboratory would spend 5 or 10 years cloning one human Mendelian disease gene. Now, it's a good graduate student project to do that. How do we do the same thing here? How do we make it simple to be able to connect these dots? Well, happily, there are lots of new ideas, lots of new data, lots of new technologies that have all come together to drive a phase 2, which is why it's the right time for the field and the right time for this community that has come together. So this phase 2 involves stretching from huge numbers of human samples all the way to disease mechanisms, from variants to phenotype, from phenotypes to variants, variants to function, function to physiology, underlain by a federated data ecosystem. Why now? You could say, well, you could always, have said this, but why now? 3 powerful driving scientific forces are the answer to why now. First, human cohorts. There are huge case collections that have been put together. Schizophrenia, heart disease. The giant cohort is itself giant, with well over 1 million people in it. These have been put together on a disease-focused basis, and as I'll come to, we're going to need those disease cohorts going forward. We're not going to be able to avoid focused disease efforts, and that's something that funders who care about particular diseases really have to keep in mind. But we also have large unselected cohorts, large biobanks. I think Decode in Iceland showed us a first example of the power of a biobank, and now we've seen the UK biobank, Estonia has a biobank, Finland, all of us. There are a growing number of biobanks, but again, I'll just keep being a broken record, not in all parts of the world by any means, and highly biased in where those biobanks are represented. But also genomics is entering healthcare. It is the case that we're going to see a lot more of genomics done in the medical system. And we do have an increasing penetration of electronic medical records, and it lays the possibility that we might be able to learn from all of that information. We expect to have tens of millions of people, but the question for us is going to be, can we actually learn from tens of millions of people? The second major trend is genomic technologies. Sequencing costs have fallen from, well, in the Human Genome Project, I think we blew $3 billion to sequence one person. That was not, as they say, sustainable. But it's now like $500 or $600 or something like that to sequence a person. And I'm quite sure that in the next 5 years, we'll see that fall to $100. Remember that $100 for people who live, if I just round 100 years, let's say, if we just kind of round it off, that's about $1 a year. That's pretty cheap. I know there are medical systems in which that's not a trivial amount, $1 a year, but for almost all medical systems in the world, $1 a year amortized is not a crazy thing. So we're not going to be limited by cost. We'll be limited more by the ability to share. Then we have single cell analysis. If we really wanted to connect all of these genetic signals to biological processes, doing it on, as Aviv Regev likes to say and will say, smoothies of mushed up tissue, is not a way to actually dissect processes, but the ability that has come along in the last couple of years to be able to do single cell analyses of RNA and chromatin at massive scale, currently tens of millions, but soon I suspect billions of cells, will give us a precision that we've never had. And then beyond that ability to observe, we have the ability to perturb. Perturbing using CRISPR-based analyses, CRISPR inhibition, CRISPR activation, with high-dimensional readouts, like from these single cells. And then finally, data. If we had to spin all these data on metal in our own institutions, we would be brought to our knees. But happily, the clouds have come in, and we have cloud-based platforms that exist. We have technologies in principle to allow secure federated data ecosystems. The question is, can we truly make them work? Let me briefly review what the powerful surprises are of what we've learned from some of this stuff that we now need to build on. Human cohorts. I am continuously surprised by the power of genotype-phenotype information. I mean, I've been a fan of it for a long time, but it's more powerful, we discover, every day than we thought. We're now discovering not just the genetic loci for any disease or phenotype, but it's not just the genetic loci for did you get a disease, but for any kind of disease-related phenotype is possible. With the right records, did you get a disease? What was the course of progression of that disease? What was the treatment response? What about family history, and how do we use that to extend the power of genetics? We can study aspects of human development, of behavior. I love the UK Biobank asking Brits, are you a morning person, or measuring a miserableness index? I just think that's fantastic that there's such a broad range of questions that are available there. But then we also know that populations have different scientific advantages. And if we're going to understand the human population, we need to we need to make use of all those different advantages. Populations that went through bottlenecks have strong alleles with stronger deleterious effects present at higher frequency. Finland has 15 million coding variants not found in the rest of Europe. And it's clear that many of them have real effects because that's what happens when populations pass through bottlenecks. The same is true about the Ashkenazi Jewish population. But in India, there are many, many endogamous populations that surely have the same thing going on, and that it hasn't been documented. What about Pacific, Oceania? We have not really looked very hard there. There are so many experiments of nature that have been carried out that contain information, and no one population will have all of those answers. Then what about recessively acting factors? There are populations that have much higher rates of consanguinity because of cultural norms than does Europe. And we can learn so much, Pakistan, India, elsewhere, and we've got to work carefully to that. And then finally, the best population of all, Africa, because it is the most diverse population of all and also has the shortest blocks of haplotypes. The haplotypes are shortest, all due to the fact that Africa is all of our home, and it has been there for longer than any other modern human population, has amazing advantages, both with regard to all this diversity above and also with regard to fine mapping, because the recombination process has been trimming things back for much longer. Now, beyond that, I'm stunned in the last several years at the power of just looking at the GWAS signal across the genome. Look at 6 million variants across the genome, each of which has been attributed an effect, a marginal effect of that. You get a vector of 6,000 effect sizes. We used to think what you were looking for were the handful of skyscrapers that crossed the threshold for genome-wide significance. But now it turns out all 6 million of these things is really interesting because you can correlate them You can correlate them between diseases. You can, I will come back to that point actually here. You can use it for finding polygenic risk scores. And these polygenic risk scores are incredibly important because beyond prediction in a clinical setting to ask who might get statins or who might get early breast cancer screening, you can ask questions like, can I study people who are predicted to have a high polygenic risk score to ask, are there processes evident in pre-symptomatic individuals? The ability to look for things that precede the disease in individuals who are more likely to get the disease is incredibly powerful in trying to separate cause from effect. So these kinds of studies. It can increase the efficiency of clinical trials because the cost of clinical trials is linearly driven by the event rate in the control group. If the event rate in the control group can be made higher by taking people who are at higher risk, you can make clinical trials more efficient. And of course, all of this requires being able to serve all population groups. It doesn't do to have great polygenic risk scores if you're a European-derived individual. The point I was making before is the other power of these genome-wide association statistics are that you can correlate them amongst each other. You can take any two phenotypes and ask, do they have genetics in common by looking at whether those vectors are the same? You can ask, are they related to a particular cell type or a tissue? Is body mass index primarily a disease of fat tissue or of brain? Well, the correlations here are more with brain with regard to gene expression patterns. Now, this is true at the level of tissues, but as we move to single cell analysis, we'll be able to do this, we hope, at finer and finer levels and ask which cell types are involved. It's still an open question. How far down can we go? Obviously, two cell types that differ only trivially by gene expression, we may not be able to separate this way, but it's really important to ask how far can we go by making use of this, and can we go beyond cell types to pathways? If I owned a catalog of all the pathways there were in biology, I could correlate it with these GWAS signals and ask which pathways seem to be implicated. Well, as I said, we have all of these different genetic resources that are being built, disease-specific and national biobanks and healthcare systems, and a distinguished author here has projected that we may have 60 million whole genome sequences in clinical care by 2025. It's Ewan Birney, he put it on bioRxiv, it must be true. I never know exactly how to trust these projections, but nonetheless, I bet it's probably about right that we're gonna have tens of millions of sequences. So whether you take the exact number right, directionally, this is unquestionably where we're going. And do we need 60 million? We do. We really do, because as we've seen, the power comes when we start getting 50,000 cases for a disease. We'd even really like to get 200,000 cases to really be able to dig deep and to be able to look at rare variants. And when you just take the UK Biobank as an indicator, and you ask, how many cases per million are in the UK Biobank, and then say, how many would I need? How large a biobank would I need to get 50,000 cases or 200,000 cases? We're gonna want tens of millions to be able to do it. Schizophrenia is a particularly hard example. There are only 700 individuals in the UK Biobank. It's underrepresented for obvious reasons, but there are real reasons, and we have to deal with that. So for all of these reasons, there is a lot to do with human cohorts. We also have the question of variance to function. We need to be able to move from genetic variants to what their functions are in the genome, and there are multiple ways to do this. By observation, by perturbation, by developing biochemical rules, by computer predictions. And I'll just say, there's a tremendous amount that is being learned from the Human Cell Atlas by documenting gene expression across every human cell type. And by doing that in enough people, 100, 1,000 people, we should be able to get all the genetic variation, common genetic variation that makes significant effects on it. And if we had that for every cell type, we would be a far ways down the line to be able to interpret these non-coding signals we see. It still won't be perfect. I know that there are activated immune cells and not activated immune cells, and context will matter. But I think we can make an awful lot of progress by moving down that road, and we should think about the human genome, the human cell atlas applied much more broadly. Perturbation. Only so much can happen by observation because evolution didn't give us all this variation. There are only a certain number, 40-ish million common genetic variants. There's tons of things we will learn by perturbation of things that didn't happen in the population. There, we can't do it for every cell type because we don't have models of every cell type we can manipulate in the laboratory, but we have many and we can learn from them by using CRISPR, for example, by using massively parallel reporter assays, by reading these things out in a high-dimensional context, by imaging, by gene expression consequences. We have attempts to develop biochemical rules. My colleague Jesse Engreitz and others have been trying to ask, could we just understand the biochemistry well enough to predict based on chromatin and contact between loci? And it turns out there are reasonably good predictions you can make already, and maybe you can do much better. And then, of course, there are machine learning approaches to ask from all of this, can we use the power of machine learning to make inferences about how all the enhancers in the genome work, to be able to predict the effect of any variant, and maybe even this comprehensive catalog of cellular proteins. Maybe that's not a complete pipe dream to try to go toward. Much of this has been generic, what I'm referring to, but we need disease-specific things as well. It'll be different what's needed for psychiatric disease and inflammatory bowel disease and heart disease, but they can inform each other. And part of this ICDA community is to have that shared learning across, because if we can figure out how to use genetics to get the biomarkers needed for disease development, or pleiotropy to detect side effects, there will be shared learnings about the methods, even if the precise assays and results will differ. And of course, we need a federated ecosystem, a data ecosystem to put it together. I've said already, we need the cloud, and we need a federated, secure ecosystem. And a lot of work is going on. I won't go into that right now because I want to turn to the work ahead. And that is what our discussion will now be. The organizing committee has been working to put together over the past several months a draft white paper. You've all received the draft white paper. The first chapter is background and describes the maps to mechanisms to medicine challenge. We're very open to input on that. That'll get rewritten. It's got to have references, but it's background. So please weigh in on all of that. The second chapter begins to sketch out the goals and what might be foundational resources that we need. And I'll briefly put them up before we turn to our discussion in a moment. But it's important to lay out those goals. It's important to lay out the things we might need. But the real work of ICDA is to figure out how we might do it. What are the proposed plans, projects, policies that would move the field forward? That's really what this community has done in the past and needs to do again. The audacious goals are easy to state. They're audacious. Know the phenotypic consequences of any variant, know the functional elements encoded in the genome and understand their functional constraints, know all the human cell types and all the cellular programs, know the processes that mediate disease development. These will, of course, sound delusional, and that's appropriate. At every point in human genetics, the things you put up sound delusional. I'm old enough to remember that sequencing the human genome sounded delusional. The estimate was it would take 2 centuries. So if you don't put up things that sound delusional, you're not trying. These are the things, and if we manage to only get 80% of the way to these audacious goals, we have no apologies. But these are the things we want to know to make this work. The white paper lays out suggestions about foundational resources for human cohorts. I won't read them all, they're in the white paper there. For about methodologies, which have been so critical. For going from variance to function, the kinds of things that we would like to have, and for mechanisms and medicine. We've tried to lay those out, and here you should be adding, objecting, subtracting, so that we think clearly about what are the foundational resources that are needed, but really, the work over the next 3 months, aiming to produce a version 1 of the white papers to talk about what would we as a community recommend as the most important first steps. You can't do everything, right? You got to start somewhere. And if we made a set of recommendations in January of 2020, we could always make more recommendations in January 2021, but we want to make the right recommendations and think about what are the right plans to do these sorts of things. So I'm going to invite Cecilia and Matt to join me, because the discussion part, the most important part of this, is to begin that discussion this morning and ask people, given this landscape that Cecilia has laid out and Matt has laid out, and I've tried to now point toward the future, what are the most important things we need to do? We'll be taking notes. We'll, of course, be writing up the notes from all of this. It won't be our last discussion on our topic, but we thought that for the very first session, we should start with a discussion on this topic. So if you guys will join me. The floor is open. What do we need to do? Okay, we win the pool. The prediction was that Manolis Thermatsakis would be the first hand up, and we won. It was not a tough, yes, the prior was pretty high.
Okay, so just to get started, before we go very much into sort of very ambitious goals, I would like to point out that there's a lot of things that we have failed in the past just because of the way we tried. Like, for example, this issue of the mix of cell types in a tissue and how this may not be that useful relative, for example, to doing single cell analysis. And I think that we need to be thinking a little bit of what can we do now much better than we could do maybe a few years ago, that if we reevaluate and reintroduce, we can learn a lot more. And I think, for example, just as an example, and that's not the only one, the aspect of looking at tissues, I don't think it's a lost cause, and I think we should still be looking at that because it's much more readily available than, for example, single cell. We have ways of dealing with it with respect to sort of doing deconvolution. There are some aspects of doing cell line analysis. So before we aim for the really, really futuristic stuff, there are some things we can achieve now that we can do better now than we could do a few years ago.
And what would they be? So I'm going to force you to be concrete. So you've made the case for don't forget about tissues.
Yes.
What would you do?
Well, I think that, first of all, GTEx has been a good example, and I think we've learned a lot. I think there are possibilities of engaging into cell line type of analysis. Now we can grow a lot more cells, and that would be often more homogeneous than looking at a piece of tissue that has a lot of heterogeneity. We can go deeper in terms of sample size into some of these cell lines. We can do a lot more difficult assays that we could do. For example, one could imagine population-based HiC in LCLs or other fibroblasts. These kinds of things have never been done, and I think these kinds of things will teach us a lot about how to see the single cell data as well as to use the sample size to learn from these mechanisms.
And would you prefer cell lines like LCLs, you said, rather than primary tissue cell lines, for instance?
No, I would prefer primary to the degree that we can, but LCLs have been extremely useful. They're not perfect, nothing is, but we just use whatever we think is relevant and has good biological information.
Great. I think we have one person, another person.
Brian Brooks, National Eye Institute. Thanks so much for a wonderful presentation, all three. I would say one of the things that we have to do is we have to think very hard in every major clinical trial that we design from basically this day forward about how we're collecting our biosamples to make sure that we can incorporate that fourth dimension that Dr. Lander pointed on of time, but also in creating, say, iPS cells that could be differentiated into whatever homogeneous cell type that you would want for a given disease. Something that we're, for instance, doing in age-related macular degeneration for retinal pigment epithelium cells, leveraging the age-related eye disease cohort, which has tremendous natural history data as well as response to therapy data.
So would it be useful to have, I don't know, at least here we're in the shadow of the NIH kind of help lay out recommendations and guidelines for what could be done with clinical trials, and ask what infrastructure is needed to help make that easier for people to do?
I think that would be great. I think it would be great also if it were an international collaboration.
Yeah, I don't know which other groups, but I think we could have multiple groups try to do that, indeed. Aviv Regev.
Aviv Regev at the Broad Institute, MIT, and for this purpose is also the Human Cell Atlas. So I had two things, but I also want to make a third point with response to Manolis. The two things are, did I catch you correctly that you felt that if 1,000 individuals were profiled at the single cell level per tissue type, we would be in good shape for the genetics community? Because I can pretty much assure you that in the HCA community, which is not really, it intersects some with human genetics, but not as much, the sense was that, as I will call it here, you people need a much larger number of individuals. And I think dispelling that would be critical in our community, because hundreds is already running now. So to get to 1,000 is actually not such a big deal if you know that that's your target. And it could shift the way that we balance between many different things that we need to balance as we design an atlas in between the two different phases. So that's point one. Point two is more of a question, both to the panel and the group as a whole. How important is the concept of modifiers and interactions for the longer term goal? Is this something that, again, as we think about technologies and analysis methods and so on, we should be worried about, or is this something we should put on the back burner as still maybe too much of a challenge or just not the right time? And the third point, in response to Manolis, a point well taken, that we shouldn't just ditch things because they feel like they're old and we should be doing the new thing. But I do want to put 2 points to consideration. The first is that it is actually becoming increasingly easier to analyze things at the single cell level, including for samples that have been preserved frozen. And so we should be very, very careful not to assume that the old methods are necessarily easier. And the second is that deriving cells is extremely difficult. It is extremely costly, and it remains costly by individual. Just the cytokines you need in order to culture your primary immune cells are a major expense and effort compared to profiling straight from the body. I see the huge value of cells as you want to perturb them and manipulate them in ways that we're not allowed and will never be allowed to do in humans, rightfully so. But I think for the basic observations, they actually carry great liabilities, and we have to be very, very careful not to build the human genetics of cell lines rather than the human genetics of the human.
Good. Those are very clear instructions to us about the sample sizes that are needed, of course, depend on the effect sizes we're looking at. And in GWAS analysis, it was iterative. We came to understand the relevant effect sizes by doing the experiments. So the question is, how can we set up that feedback between the single cell community and the genetics community, that we can begin to home in on what are the right sizes to detect the right effects, what are the right effects that are relevant to disease? And I think there's a really rich topic there.
So we have done, I don't think this microphone is on.
Is that on?
I don't know.
Yeah.
It's on. So we have done preliminary power calculations together with Ben Neal, just looking at some of the effect size ranges. And initially looking at the low-hanging fruit, knowing we had winner's curse, I think we ended up on 700 to 800 samples done in duplicate. But when we have done computer vision analysis paired with machine learning to extract cellular phenotypings out of the histology samples in 1,300 individuals, we can't find any genetic associations that are robust there. So that gives you a flavor for the V2F that you're going to have to scale up significantly. So that's work I'm doing with Melina Klausnitzer and others. So we're scaling up to 5,000 individuals now just to see, because it's much faster to look at histology and extrapolate than to start a culture in the lab, for instance.
Yes.
I was going to come in on that because I think there's a couple of interesting points there. One is the sample size issue, I think, will vary considerably from disease and tissue to disease and tissue. And I think one of the key things is going to be the degree to which those regulatory effects are cell type specific versus context specific. And there will be some inherently context specific systems like blood, where that may be more challenging than others, which may have less context dependency. So I think that's going to be a key issue. I don't think we know the answers yet.
Yeah, I would strongly say that in disease you have a lot of context specificity of the regulation. Some of it has to be the disease tissue, and some of it is probably just the genetic background of the individual, and today we actually don't distinguish those so well.
Right. But of course the key is, how do we learn, right? We can all guess, but what's worked in the past is a layered series of projects that teach us the empirical answer. So, yes.
Okay, Mila Prakonina Olsen,
Oops.
NCI. Mila Prakonina Olsen, NCI. My point is that a disease is often or always a product of gene-environment interactions. And what we are measuring is steady-state conditions when the causing factor is maybe well gone long time ago. We are just reading an output of that. So we need to figure out how to better model those conditions where disease is initiated. And those can be transient, so we need to understand biology to better model it in vitro and possibly then in vivo, and expose our cells or primary cells or cell lines to those factors and measure those changes and correlate with genetics.
Great, thank you. Eric?
So thank all three of you for setting the stage. Eric Borwinkle from the University of Texas and Baylor College of Medicine. I guess 3 comments, and it's along the lines actually of the previous discussions. The first is not to forget about measuring the environment. You know, Eric talked about, you know, the cost of getting a genome, and it's going down, and maybe it's going to be $100 in a few years. I actually think this group in particular, we were handpicked. You know, we're quite comfortable with that, and we're quite comfortable with, you know, the challenges of the data. But my prediction is that pales in comparison to the difficulties of measuring the environment on walking, talking human beings. And I think if this is going to be successful, we need to rise to that challenge and not ignore that. And then along those lines is many of the diseases on the 3 of your slides are late onset diseases, and we need to understand how health gives way to disease, that process. And you know, for example, that the challenge there is we need to think about following people longitudinally, whether that's in the population or in the healthcare system, often depends on where we sit. It's a bit challenging in the US, as you know. And then along the lines, actually, of the previous discussion of cells and tissues, again, we need to look at those throughout the lifespan and healthy individuals, because the disease itself is going to drive many of those processes and make the genetic effects pale in comparison. And likewise, Eric, in your slide that I think the samples on the left, my left from the audience, and disease on the right, once again, at each of those steps, we need to have ethnic heterogeneity and ethnic representation. It's not good enough just to sequence a large number of people from many, many populations, but yet all of our cell line work, for example, or tissue work is done in populations of European descent. We need to put the whole package together across the spectrum.
Indeed, I think to great credit, the HCA, the Human Cell Atlas, has focused on the fact that the cells can't just be European cells.
I wanted to push you and ask if you have ideas on how we measure the environment sort of for the walking, talking, breathing human being, because it's such an interesting concept, isn't it? But so challenging, so it's exciting.
Yeah, but you know, I forgot which one of you said it. Just because it's really hard, we can't run from it. In fact, because it's really, really hard, that's what we should be focusing on.
Yes, but I was curious.
And I think it's going to be a combination of very traditional things, of you know, asking people questions, whether it's, I forgot what the example was, whether you're a morning person or not, is do you like bananas? And the other is, you know, there's an adage, we are what we eat. And we probably need to think about more biochemical readouts of the environment, whether they're epigenetic or whether they're, you know, molecules circulating in the bloodstream or other tissues. But I do think as we come together, we don't just congratulate ourselves. We really have some major challenges, I think, to bring together genes and environment. And for these common diseases, I would contend we need to do that head on.
No, this meeting is about the challenges. I think we're kind of done with the congratulating part. It's all about the challenges, but we're going to have to reduce those challenges to concrete projects, because we got to at least start. And so directionally, yes. Now, what do we do? I'm going to go back here, and then we'll work our way around the microscopes. Yes?
Phil Smith from NIDDK. So one of the things we're forgetting here is the developmental context of disease susceptibility. And so, for example, one could be born with less nephrons. One could be born with less islets. One could be born with less arcuate neurons. All of those things could be entirely normal looking, but because of their number is low, make them susceptible to environmental stress and thus disease. And so we don't have fundamental knowledge about human anatomy and biology that we can get. And that's relatively not the same scale, but it's stuff we don't have. We need to do more of it.
And do you know what you would do if it was just up to you or TDK? What would you do?
Yeah, so I would actually try to collect every possible newborn or fetal, sample that becomes available, and I would do analysis of disease, of organ structure across the entire organism to get some sense of what is the variation in the number of islets. I know in the islet, in the diabetes community, we know there are differences, and so we don't really know how that correlates to disease because the sample size is very small. But we're building that library. We need to do that across tissues of all kinds to get a sense of just how much the variability in structure might actually make us susceptible to disease.
It'll be exciting in the NIH discussion later today to hear broadly about the thoughts across the NIH along these and other directions. Let's go here.
Hi, Rick White. Hello? Rick
Yep.
Wychick from NIEHS. I just want to reinforce the whole environmental theme. And if you want to ask how to do this, actually my colleagues at the Institute have been doing this for decades. So there are systematic ways of collecting information. But I could propose that one of the first things we need to do is pay attention to patient-physician interactions. So physicians should ask their patients, where do you work? What are your types of exposures? What neighborhoods do you live in? Are there high pollution and asthma? I mean, there are so many different correlations between environmental triggers and different diseases. And unless we have this information in the health records, we're not going to be able to make the connections. So the other thing, too, is we have to pay attention to the developmental origins. So the environmental exposures during pregnancy, and in fact, I was just at a meeting even before pregnancy, whether fathers are smoking can actually predispose whether or not children will have deleterious health effects. So the environment is a biggie. During the panel discussion this afternoon, I'll reinforce this. And we also want to focus on public health. It's not just about medicine.
It would be very interesting to hear NIEHS ideas on what kind of best practices can be adopted by cohorts and biobanks going forward, especially using new modalities, cell phones to gather data and other things, because it'd be great if similar questions were being proposed, and not just NIEHS, right. Funders in other countries as well, but I pick on you because you're here.
Sure. I mean, we've had the Gene by Environment initiative in the past, and NIEHS has participated with other ICs across the NIH on how do we collect this information. I mean, NIEHS is a lot of it is about toxicology, but other institutes are very interested in the environment, psychosocial stresses. So developing the mechanisms to do this, and actually one of the new programs at NIEHS is something we call the Exposome, which is a way of attempting to begin to evaluate the totality of exposures. So we've been funding projects to develop the types of wearable sensors so that you know what type of PM 2.5 you're exposed to. So you have good solid data to correlate that with genetics resources.
I was just going to say, well, I think one of the real opportunities is to harness that kind of long-term expertise in quantifying the environment with these kind of newer genetic approaches of predicting environments, especially developmental environments. So I think there's somewhat alluded to that as the opportunity to use genetics to actually explore the environmental space.
So we're very excited about this.
Yeah. Rory?
Rory Collins, Oxford. In Matt's talk, he mentioned the opportunities of personalized medicines and giving a patient the right intervention. I think also we need to be concerned that we don't withhold inadvertently the right intervention from patients, and to think about the asymmetry between the likely genetic impact on side effects compared with main effects, because certainly in cardiovascular disease, I've seen a lot of remarkably naive genetic associations in terms of effect of treatment, claiming differential effects, which I think well, I've shown in some cases, and I think are likely to be false. So I think that asymmetry is really important to think about.
Hillary?
Hillary Finucane, Broad Institute.
Talk loud.
Hillary Finucane, Broad Institute. Is this on? Yes. Okay. And Matt's talk, he pointed out that a lot of what we've been able to do with summary statistics. And I wanted to highlight that a lot of that has been summary statistics with a matched LD reference panel, which is something that we don't have for a lot of populations. And so as we move towards diversity, there's a lot that we won't be able to do without having either in-sample LD or some very thoughtfully constructed high-quality LD reference panel.
Yeah, so we shouldn't leave here without some very concrete things on paper that don't just say we need characterization of many populations or whatever. We should leave with we need N individuals analyzed in the following way from each population of a given sort, because exactly the clarity of what do you need from that will help drive projects.
To me, it's also a question whether it can be done with reference panels or whether we'll actually need random subsamples of the studies themselves.
Cool.
Hi, Isia Lopes-Senders from the Brazilian Initiative on Precision Medicine. And I have a comment about diversity. Because when I hear that there is an overrepresentation of European populations in the different studies and databases so far, we have the impression that Europe is homogeneous, and we know very well it's not. So I think there is underrepresentation of Southern European populations in these different studies and databases. This is very important from Latin America. So I think this is something that should be addressed as well. And I have a second comment. It's about the benefits of admixed populations for some of the strategies of mapping complex disorders. So I think that's also something that it's important.
Great. Cody.
When we are thinking about the environmental exposure, I think it is very important to recognize that we inherit compulsion to seek environment and avoid environment. So I think that probably the biggest key to the understanding of the environmental influence will come through developing understanding of how the brain works. Use genetics to develop understanding of how we think, how we feel, why we feel, because thoughts and emotions govern behavior, and the behavior is what dictates the environmental exposure. So just keep in mind, there is no line of distinction between nature and nurture.
Yeah.
It doesn't exist. It's an illusion.
Very true.
Hi, Kathy Klinger from Santa Fe. So,
no, talk like right into the thing, then it gets loud.
It's a good thing, and I will tell you I'm widely known at work for being the cause of people needing hearing aids. So I shall try very hard. So, one of the things I don't want us to, as we focus on how much the cost of generating genetic data, sequence data, et cetera, is falling, I don't want to forget the phenotypes, right? Because we really are particularly thinking about chronic diseases that occur over long periods of time. The phenotypes that are in the healthcare records, the phenotypes we're used to collecting, are disease-driven. They're in-stage, or at least clinical representation stage-driven. So if you think about even a simple monogenic disease like polycystic kidney disease, by the time the things we measure in healthcare, like GFR, are declining, the story's over. 80% of your renal function's lost. Right. So and that's a trivial, easy example, and that's one where, in fact, the NIH helped fund a lot of work on sequential MRI to generate other indicators of disease progression. And so as we think about these much more complicated systems, to be thinking about, well, what would we want to measure instead? And how are we going to fund validating those measurements, establishing those measurements, disseminating the ability to collect those measurements so that we have the phenotypes, not just are you a morning person, which is important, I'm not, but the phenotypes around the actual physiological processes that are going on that we might want. And the second part of that is, at least in the US healthcare system, we actually collect a lot of data that never gets reported into the healthcare record because they're automated systems that collect. It's cheaper to run all of the assays, but you only report out what you've been requested to report out. And so there's a lot of data actually that could be unpacked, that the cost of generating that data is already there if we only could find a way to unleash it. So I just don't want us to forget those things when we think about how do we progress going forward, because they're concrete, they're actionable.
Great. We're just going to take two last questions because I want to keep us on time. I think you were up next.
Jonathan Pollack from the National Institute on Drug Abuse. So I think one of the big obstacles here is basically data integration. So I think there are standards called FAIR, which is findable, accessible, interoperable, and reproducible. So to have something findable, it has to be a machine has to be able to see it. So something like RRIDs or another system where data is labeled, something that is accessible so that you know what the actual standards are, that you can attribute and give credit to that, so it becomes something like a science citation index. And interoperable, we really need APIs that would integrate all the data, and it needs to be reproducible. So
that's great. All right, we'll take two questions there, because I see Lou there. So, yes.
Hi, Caroline Fox, Merck Research Labs in Boston. I just wanted to give the pharmaceutical drug development perspective, because we ultimately are the end users in all of this. And for us, the most important thing in this entire ecosystem is really elucidating mechanism from the genotype-phenotype associations, because that's the only way that we can really understand how something works and then think about developing a drug against it. But that also directly leads to sort of your second aim, which is patient enrichment and stratification, because once you know the mechanism, then figuring out if stratification or enrichment in a clinical trial is going to be necessary is an obvious next step. And just sort of looking to the future in healthcare and payers and trying to get the right patient, the right drug at the right time, and not wasting money in the healthcare system, that's going to be absolutely essential. So I just wanted to sort of lend that perspective to the discussion.
And I think an important part of the ICDA's formation is the recognition that the pharmaceutical industry and the biotech industry are very much a part of this community, and that we need knowledgeable people from the industry to say, what projects do you need to supply the kind of knowledge that will make it easier to choose good targets, to be able to develop biomarkers, to be able to stratify patients, but most importantly, to be able to saturate mechanism and know that you have a real mechanism that you can use pleiotropy to look for potential side effects. I think there's a whole rich body of work that we haven't thought as much about what does genetics mean in the development of therapies.
But also, as was pointed out from our colleagues from NIH, like keeping public health close at heart, because it's going to be really important to sort of disseminate the information into preventive measures on a population base, I think.
Right. Lou Stout gets the last question.
So maybe I didn't hear the word cancer too much. I am from the National Cancer Institute, and it's probably because it's a tough topic. And it is tough because of the heterogeneity that is profound. So I'm not saying that it's scalable to do a deep dive into every human's cancer, but I'm going to advocate for having the ability to do so if circumstances and resources change. So being able to identify the piece of cancer tissue that's attached to a human being that's part of one of these cohorts, to then be able to reach to that and use all the rich knowledge we have of somatic genetics to help us tease out the causative variants.
So why don't you want to say we should just do that? You seemed reluctant to say what I believe you believe.
It hasn't been done, to my knowledge, it's not being done in all of us. It's maybe Eric will correct me, but it's expensive to do that. There are really good ways. In the United States, we have the SEER program that we in the Cancer Institute are expanding into, so that we can now not just have the ultimate outcome for the patient, live or dead, but actually where that piece of pathology tissue is. So we're working on it. And I'm just suggesting an international scope of this because of the heterogeneity of cancer requires it.
I think if you've got protocols in place for how to do that in biobanks, this is an excellent opportunity, given the representation, to actually sell that idea. I think that there would be a lot of buy-in. So I think that sounds like a great project to propose, and something that one could start with.
Yeah, look, the point of this whole discussion these 2 days is to think about what can we be doing now. Yeah. And I think Yes, it'll take work to do that, but we might as well put it on the table now and define it well enough. The challenge often is not directional. The challenge is actually making something concrete enough we can do it. I know how hard NCI has been working in this direction, and I think this would be a great time to really deepen the conversation between the germline genetics community and this largely somatic-driven cancer community to ask, once we have tissues coming into play, why aren't we doing this in every single case? And also taking advantage of the records that are kept about progression and response. So very much in bounds for this.
And again, just quickly, because people have been talking about interaction analysis, so gene-gene interaction analysis between somatic mutations in tumors and the germline is something that I don't work in cancer genetics, but would be super excited to see.
Super. So keeping us on time, we're going to stop now. I'm not going to thank the speakers because I was a speaker, but I'll thank those speakers.
Thank you.
and thank all of you for engaging the discussion. We will break and resume at 10:17. You get back 2 extra minutes.
Perfect.
That's why I called up Lou, because I realized that I did that.